Now we're going to talk about cell signaling for taste and smell. And we're going to begin with some terms. The first is anosmia, which is the loss of the sense of smell, aguesia, which is the loss of the sense of taste, tastins, which are the chemical compounds that bind the taste bud receptor cells and impart the primary flavor categories, of which there are 5: sweet, salty, bitter, sour, and umami. There's also a kind of proposed 6th flavor category for fat tastants. However, the addition of a 6th flavor category is still relatively controversial. And then odorants. Odorants are the chemical compounds that bind to and activate the odorant receptors and impart a sense of odor as a result. And we will begin with the signaling cascade for taste cells. Taste cells are comprised of the taste buds that are embedded in and expressed by specialized epithelial cells, particularly in their cell membrane on the apical domain. And in this picture on the right, this top cell represents the taste cell. The apical domain is up here at the top of the image, and then the basolateral domain is beneath it. Now, to orient you further to this image, the taste bud cell is the top cell, and then the primary sensory neuron is beneath it. The synapse between the epithelial cell and the primary sensory neuron is this gap right here in between the two cells. This would then therefore be the peripheral terminal of that primary sensory neuron. Now, in the apical domain, there are a number of things that are needed to impart chemosensory transduction. There are microvilli, which are not particularly expressed in this particular image here. They're not particularly shown. There are tastant receptors, which in this case is this purple receptor shape right here. And then there are voltage-gated ion channels, which are over here. And then there are also TRP receptors. TRP receptors are kind of all over in this particular image. There's a TRPM5 receptor, which is expressed down here on this particular image, but they are, again, all over. And then within the basolateral domain, at the bottom of the cell, there are a number of more ion channels for the different cations. And then there are the vesicles containing neurotransmitter, which is this circle right here, and these green dots are representing the neurotransmitter itself. And the neurotransmitter would be released into essentially this cleft between the epithelial cell and the primary sensory neuron. The neurotransmitter could be bound by the neurotransmitter receptor expressed by the primary sensory neuron to induce a change in electrical potential. And should that change in electrical potential be sufficient, you can generate an action potential in this neuron. So let's talk about the signaling cascade just in big, broad terms. What initially happens is you have a receptor that is going to bind a flavor tastant. And that could be any of the main flavor tastants that we talked about previously. If it is a flavor tastant that is comprised of salt or acids, that would be essentially acid or hydrogen ions or salts like sodium ions, then that would be transmitted through an ion channel. So that differentiates the initial tastant binding signal cascade, where a sweet, bitter, or umami flavor is going to bind a G protein coupled receptor, whereas the salt or acid flavors are going to flow in through an ion channel. Regardless of what happens, there is an internal signaling cascade. If it's a G protein coupled receptor, that goes through second messenger processes. And if it is simply an influx of positive cations, then either way, you're going to end up with a depolarization in this epithelial cell. That depolarization will trigger additional cation fluxes, whether it's influx of sodium and a concurrent efflux of potassium, further influx of calcium ions. Regardless, that increase of calcium ion concentration within the intracellular compartment triggers the binding of these vesicles to the basolateral domain and then the release of their stored neurotransmitter. If that stored neurotransmitter in this example is serotonin, it would bind the serotonin receptor expressed on the peripheral terminal of this primary sensory afferent neuron, and again could, if sufficient enough, trigger an action potential, which would then be transmitted on toward the brain. Now, as you saw in the previous graphic, there are a lot of intracellular signaling cascades that are involved in this process. This is broken down here into the basic components. It starts with a ligand binding a receptor on the apical domain. However, in the case of sour or salty or hydrogen and sodium, that would influx through their sodium or hydrogen ion channels. That will trigger a change in receptor potential in the taste cells, and these are usually depolarizing electrical changes. That depolarization triggers voltage-gated calcium channels. Voltage-gated calcium channels open when there's a change in voltage, and that is induced by the depolarization. That triggers the transmitter release. The neurotransmitter involved in the mechanism of its relief will differ by cell type. There are two different types of cell signaling cascades that will trigger either the release of ATP or serotonin. And that was shown on the previous slide. Now you might say to yourself, okay, well, serotonin makes sense, but how is ATP a neurotransmitter? And we think of ATP most classically in its role in energy processes and metabolism, but you may not have been aware of this, but there are neurons that express receptors that will in fact bind ATP. And when that occurs, the binding of ATP on the cell membrane with its particular receptor will trigger the similar sort of intracellular signaling cascades induced by other classical neurotransmitters. And when that happens, the neurotransmitter binding to its receptor on the primary sensory afferent peripheral terminal will generate a receptor potential change that, if it is sufficient enough, again, will trigger an action potential that can initiate the signaling cascade that ends up going to the brain. Now, this is the breakdown of the tastants by their flavor profiles and then their associated stimulus that comes from them, and then the neurotransmitter that is released by the cell type that that particular tastant would bind to. So in the case of, for example, sour tastants, which is primarily derived from the acids in things like citrus fruits, that stimulus is hydrogen ions, and the neurotransmitter released is serotonin. For salty, that is primarily derived from sodium chloride or sodium alone, which are binding epithelial sodium channels, or its generic ion channel, again releasing serotonin. And you'll notice that there is a commonality that the cations, whether it's hydrogen or sodium, end up releasing serotonin as their neurotransmitter. So that is their commonality. For sweet, of course, that is coming from sugars. Sugars bind G protein coupled receptors, and the epithelial cell that expresses these and are activated by sweet receptors release ATP. Umami is derived from amino acids, particularly in meat and other sorts of similar amino acid containing compounds. The biggest amino acid of note is glutamate, which binds a specific metabotropic glutamate receptor, mGlu-R4. Now, don't forget that there is a mGluR6 where, and that is in the eye. That is important in binding, particularly in terms of the phototransduction pathway in the retina. So this is a G protein coupled receptor, and again releases ATP. For bitter compounds are found in things like coffee and bitter green leafy compounds like kale or something like that. There are a variety of different compound structures that make up bitter taste profiles, and they also bind G protein coupled receptors and release ATP. So as you see here, when we have cation channels that are involved, we release serotonin. When G protein coupled receptors are involved, we release ATP. Bitter taste receptors are particularly important to the human experience, and that is because bitter taste is generally associated with a lot of compounds in nature that can be poisonous or toxic. And humans have evolved to discern bitter taste as an aversive experience and something to avoid. And it's thought that that is meant to be a mechanism by which we can innately guard against consuming things that might be harmful to us. And the process by which this occurred is through the G protein coupled receptors that bind bitter compounds. They bind those compounds with such high binding affinity that as a result, it takes just a very low concentration of a bitter compound or a chemical composition of a bitter substance in order to detect it and essentially taste it. And that is helpful because since you can discern a bitter or aversive taste at such a low concentration, hopefully the individual would sense that bitter taste and avoid the additional ingestion of further potentially toxic substances before it can make them sick or have a fatal result. However, of course, as we all know, some bitter compounds are poisonous and toxic, and many are not. And in fact, people have learned to tolerate or even seek out various bitter and other aversive compounds, even sour tasting compounds, because as a human, we can learn to enjoy them or appreciate it. Caffeine, its chemical structure, is discerned as a bitter compound, and yet we as humans tend to seek it out. We also tend to mask the bitter taste of caffeine by adding things like milk and sugar to our coffee. Green leafy vegetables contain bitter composition compounds, and so on and so forth. So essentially, the human experience can overcome those innate taste responses by learning what is and isn't toxic, and essentially consuming the stuff that is okay for you and avoiding the stuff that will make you sick. Now we're going to shift gears and talk about the cell signaling cascades involved in odorant receptor neurons. Odorant receptor neurons can be abbreviated to ORN, so you'll see that I use ORN a lot throughout the rest of this lecture. Odorant receptors are particular receptors expressed in the cell membrane of the primary neurons that transmit odor profiles. Humans have about 350 different odorant receptors that we have identified. So, I mean, that sounds like a lot, 350, but those are 350 very specific different odorant receptors that respond to different odors. Now, compare that to the fact that humans have about 12 million neurons that express odorant receptors. Compare that to, say, another lower mammal species like a rat, which has about 15 million odorant receptor neurons. But strikingly, look at something like a bloodhound, the dogs that are famous for their amazing sense of smell. Look at how many odorant receptor neurons they express. About 4 billion. I mean, no wonder they can smell so many different things compared to a human and even to a rat. So olfactory neurons, they're exquisitely sensitive to odorants. And of note, olfactory cells, so the olfactory neurons, they are specialized bipolar neurons. They use glutamate as their neurotransmitter. And in this image on the right here, we're showing you an example of 3 in this kind of dark pink color bipolar neurons. These are the odorant receptor neurons, or the odorant neurons. Their receptors would be expressed down here at the bottom of these neurons. And then in between them, there are supporting cells, which kind of make up the area in between all of these bipolar neurons. And they are either called support cells or by their other classic name, which are basal cells. Together, they make up the olfactory mucosa, and their basolateral domain abuts the basement membrane. Now, since these are bipolar neurons, they don't really look like bipolar neurons just in this graphic, but they are. That means that they have an axon coming off of their central terminal, which is going to end up going toward the brain. So I just got done saying that humans only express about 350 different type of odorant receptors. So then how is it that humans can detect something like 12 million different smells if we only have 350 different types of odorant receptors? Well, that's because a given odorant, so one chemical compound that we can sense as an odor, can stimulate more than one type of odorant receptor. And then the combinations of the different odorant receptors that are stimulated act kind of like a signature of that odorant that you then learn to identify with that compound. So let's look down here in this example. We have hypothetical 8 different odorant receptors. Remember, humans have about 350, but I didn't have enough room on this slide to show 350 different boxes to represent the odorant receptors. So we're only showing you 8 different ones. Now, say the compound in a rose, a flower that you smell and you can immediately identify as a rose, that particular chemical might activate 4 different odorant receptors, and the combination of receptor 2, 4, 5, and 8 is transmitted as a sense of smell of a rose. And if you were to say eliminate this hypothetical odorant receptor 8, and you only activated receptor 2, 4, and 5, then you may not smell a rose at all. You may smell something completely different. So let me erase these and compare that with the binding of a chemical from sweat, and the sweat chemical might bind odorant receptor 1, 3, and 7, and that combination tells you it's sweat. But if you were to eliminate the binding to odorant receptor 7, you might get a completely different sense of smell. In addition, the concentration of the odorant also matters. So I just got done talking about if you have different combinations of odorant receptors that are activated, you get different smells. However, the concentration also changes what you might smell. And this is an example here. This is the chemical compound for indole. Indole is found in some flowers. And if you only have a very low concentration of indole, and I don't really know what the millimolar range would constitute, but at low concentrations, it only binds one odorant receptor, and it smells quite pleasant. It smells like flowers, which is why there's a floral smell over here. But at very high concentrations, and again, I don't know what the millimolar concentration is, but it has to be quite many-fold higher of the same exact chemical compound. It will bind more receptors. In this case, it binds 4 or 5 different receptors, and this smells quite aversive. It smells putrid, and it's very unpleasant. So again, the exact same chemical compound can transmit 2 very different senses of smell just based on its sheer concentration. So combining all of that, the types and combinations of odorant receptors that are bound by a given chemical compound, and then the concentration of that compound is what gives us the ability to discern something like millions and millions of different smells, even though we only have 350 different odorant receptors. So now let's talk about the sensory chemotransduction cascade in odorant receptor neurons. This is a very big picture view, and the next slide is going to show you a graphic that depicts all of this information. So step one, we have an odorant or a chemical compound that diffuses into the nasal mucus, gains access to the receptor that it is going to bind, which are expressed on the cilia of the olfactory neuron, and then activates that olfactory neuron cell. The specific receptor protein that binds odorants is a specialized G protein coupled receptor called G-ULF receptor or GULF receptors, just like the game. Like other G protein coupled receptors, it does use a second messenger transduction system to trigger its intracellular signaling cascade. In this case, it is cyclic AMP. Cyclic AMP opens a special channel called cyclic nucleotide gated channels, CNGCs. These are gated channels expressed in the receptor that are opened by cyclic nucleotide compounds like cyclic AMP. When these gated channels open, you are going to allow cations, particularly sodium and calcium, to influx into the cell. And what happens when you have cations influxing into a cell? Well, usually you have a depolarization that occurs. When depolarization occurs, we have additional channels that opens. In this case, there are chloride channels that open, and that will provide the remainder of the depolarization needed to generate an appropriate receptor potential that at sufficient quantities can achieve an action potential. These chloride channels are called calcium-gated chloride channels. Okay, so what this means is the calcium that influxed as a result of these CNGC channels, that calcium suddenly appearing in higher concentrations on the intracellular side of the membrane activates calcium-gated chloride channels. Okay, so I know that was a lot of steps and you're probably really confused right now. So let's look at the graphic on the next slide and see if that helps a little bit. All right, so here is a rather busy looking graphic, but we're going to break it down step by step here. So step one is up here. We have an odorant molecule, which is this pink looking hexagon that is going to bind to its receptor, which is expressed in the olfactory receptor neuron. So this is the olfactory receptor neuron down here, O-R-N. This receptor is going to activate a G protein initiated signaling cascade, okay, because it is a GPCR. This is your GPCR. And then if you're familiar with some molecular biology, you might recognize this beta gamma subunit. I'm going to skip over that and just kind of call it physiology magic for now. But you'll see that it initiates this G olfactory neuron signaling cascade, which is right here. So this GPCR is the G-ULF or the GULF, okay? When that occurs, you have a second messenger signaling cascade initiated through cyclic AMP. Cyclic AMP is a common and famous second messenger signaling cascade initiated by a variety of G protein coupled receptors, or in this case, the G-ULFs. Now, cyclic AMP is now abundant and flowing around and floating around on the intracellular side of this membrane. And since it's floating around, it's going to eventually encounter a cyclic AMP binding site. That binding site is right here, and this is the cyclic AMP gated channels. So up here are the cyclic AMP nucleotide gated channels, so the CNGCs. That's what we were talking about on the previous slide. Upon the binding of cyclic AMP to its binding site, this ion channel opens, which allows calcium and sodium to influx, okay? So now we're adding a whole bunch of positive signal into the interior of the cell membrane. We're adding a bunch of positive ions. So what happens when you add a bunch of positive ions to the inside of a cell? Well, its overall electrical potential, it goes up, it changes, okay? Now, because we have a whole bunch of calcium floating around inside this particular odorant neuron's intracellular membrane compartment, we have a bunch of calcium floating around. There's a bunch of more physiology magic that occurs. And at this point, it gets really complicated. And I'm not going to make you memorize the exact, the fact that there's an exchanger and there's all these different things happening. The big takeaway here is calcium triggers this channel and this channel. And together, the combination where we open up these chloride channels, this is your calcium-gated chloride channel here. So there's a binding site right here for this chloride channel. And the calcium binding to it opens up this pore. And when this pore opens, any floating around chloride is allowed to escape. Okay, so now we are losing negative ions. And in addition, there's some fluxes between sodium, and there are fluxes with calcium. And the net result is a depolarization, or an increase in the positive charge on the interior side of this cell membrane. Okay, so our electrical potential has now changed such that the net result allows for a depolarization of this neuron. Okay, so big takeaways. Definitely know this G protein coupled receptor uses cyclic AMP as its second messenger system, which binds a cyclic AMP gated, number 3 here, a cyclic AMP nucleotide gated channel, which then opens a variety of calcium-gated ion channels, particularly calcium-gated chloride channels, that results in a depolarization of the membrane. And I'm going to make sure that these show up really well. So number 1 is up here. Number 2, second messengers with cyclic AMP. Number 3, cyclic nucleotide gated channels. Number 4, calcium gated ion channels. And then number 5, depolarization. Okay, so now that we have depolarized our neuron, what happens when that odorant compound just hangs out, and after a while, essentially what happens? Well, if you've ever noticed you've been somewhere and a smell, particularly a pungent smell, is very strong, eventually you get to what you get used to the smell, and you seem to either think it's no longer as strong as it once was, or you don't really notice it. Well, this is an actual physiologic occurrence and called adaptations. So the odorant receptor neuron literally adapts to the chemical compound the longer it sticks around and binds to the receptor. And there are essentially 3 possible mechanisms that can contribute to this. The first is the receptor potential is reduced in magnitude because cyclic AMP concentration can decrease. Cyclic AMP gets broken down enzymatically the longer it sticks around in that intracellular compartment. And so if you don't have enough cyclic AMP around, or it's not as much as you used to, then you're going to have less binding to its binding site on those CNGC channels. Second, there is a recovery process that occurs because the binding of calcium to its calcium signaling cascade. This is part of that physiologic magic that I showed you in the last slide. Calcium binding to its signaling cascade can have a reduced affinity for the channel to cyclic AMP. So that calcium calmodulating signaling cascade and the cyclic AMP binding site for its ion channel has a reduced affinity eventually to cyclic AMP. So cyclic AMP can get broken down, and then the affinity for cyclic AMP decreases. And then finally, back up at the very, very first step, which was the odorant receptor molecule, receptor itself can become phosphorylated. So this is the G protein coupled receptor that is specialized and called that G olfactory receptor, that G-olf receptor, that can become phosphorylated. So you have your cell membrane here, and this is your G protein coupled receptor. Essentially what happens is you start sticking a bunch of phosphates on it, and this is a pretty common mechanism in cell biology and physiology and molecular biology, whereby if you start phosphorylating stuff, it changes its sensitivity. And in this case, if you phosphorylate this receptor with a bunch of phosphates, it changes its ability to react to, bind, or change its sensitivity to the odorants, such that it's just simply going to take a greater concentration to activate this receptor than ever before, because it's now got a bunch of these phosphates stuck on it. So 3 mechanisms. We have the enzymatic breakdown of cyclic AMP, reduced affinity for cyclic AMP to the calcium-gated ion channel, and the cyclic AMP gated nucleotide channel, so both. And then the odorant receptor can become phosphorylated, which modifies its sensitivity. So here is a big picture summary of the odorant signaling mechanisms. We have G olfactory receptors that stimulate cyclic AMP, which opens those cyclic nucleotide gated channels that leads to cation influx and eventually depolarization in the membrane. And then as you get used to a smell, there's 3 things that can happen. You have a decreased sensitivity for cyclic nucleotide gated channels to cyclic AMP. Cyclic AMP can be broken down, or the receptor can be inactivated or desensitized by phosphorylation. So that concludes this first learning objective. Now, when it comes to anosmia, or reversible hyposmia, the most common cause is having a cold. Essentially, having very thickened mucus will block the ability of the odorants or the chemical compounds that would bind to an odorant receptor to get access to the receptor. And so therefore you have a decreased sense of smell. And this is an example of reversible hyposmia. And another cause of anosmia or reversible hyposmia is damage to the olfactory epithelium itself. So we're very particular when it comes to hearing loss and vision loss, and we take public health measures to protect those. But we do forget that olfaction loss is very possible. And as a result of a loss of smell, you can have a diminished loss of taste or a diminish in the ability to taste. And that loss of tasteability, like we talked about with the decline in aging, can have an impact on your ability or desire to eat and change the sort of ways that you season your food. And of course, if you were to add lots and lots of extra salt to kind of compensate for loss of taste as it arises from damage to the olfactory epithelium, that could be a problem, particularly in someone with hypertension. Head trauma can damage the olfactory epithelium, particularly damage to the cribriform plate, which can physically shear off the olfactory bulb as well. In contrast, hyperosmia, or increased sense of smell, is a physiologic phenomenon commonly known to occur in relation to migraine, psychotic states, and even during pregnancy. Now, just something to note that neurogenesis, or the birth of new neurons, in the adult brain only occurs in 2 places in the entire brain. Otherwise, the neurons that you're born with are essentially those that you're going to have, that's all you're going to get. But when it comes to the olfactory bulb and then in the hippocampus, these are the only places that neurogenesis is known to occur in the entire brain. And finally, when it comes to neurodegenerative diseases, tracking olfactory senses is actually becoming more and more important. There exists an abundance of evidence in the literature that impairment in olfactory senses is a common early finding of various neurodegenerative diseases. In fact, in Parkinson's disease, for instance, neurons of the olfactory system are among the first to demonstrate pathology, even years sometimes before motor deficits are manifested. And therefore, work is underway to try to utilize olfactory testing as an early diagnostic biomarker for various neurodegenerative diseases, as well as used to try to predict the clinical outcomes or severity of those diseases before they manifest in order to try to get early intervention. Of course, you can't talk about anosmia and the impact of the nervous system because of changes in olfaction and taste without thinking of the kind of famous symptoms that have arisen from the COVID virus. I'm not going to address that in this lecture series, but am going to try to come up with a short amount of information because I know there's a lot of interest on your behalf as to what is the underlying cause of anosmia and aguesia with that virus. So look for that separately. That concludes this lecture preview. We will see you in formal lecture in class soon. Thank you. We know that loss of taste and smell are hallmark symptoms of COVID-19. However, what we don't know are the underlying mechanisms that contribute to this loss of taste and smell, either completely or even partially. When it comes to loss of smell, there are three kind of major hypotheses in the literature with some evidence to support them. The first one has really been kind of dismissed by this point. A year ago, it was still being considered. By this point, we've really dismissed this idea of physical blockade of the olfactory neuron receptors due to nasal congestion. And that's really because we just don't see a lot of nasal congestion with COVID-19, and yet there can still be complete anosmia. More likely is a disruption in what we believe is the extracellular environment around the olfactory receptor neurons due to infection of support cells known as sustentacular cells. Sustentacular cells have been known to be infected by COVID-19 viral particles, and they can die off as a result. Importantly, sustentacular cells are also regulators of the extracellular environment in terms of electrolytes, and particularly related to ions, sodium, potassium, balance, etc., stuff like that. And if you disrupt that extracellular environment, especially in the context of neurons and neuron signaling, because that's what olfactory afferents are, or neurons, then that can really change the function of these olfactory neurons and how they process information. The third hypothesis has to do with just a kind of a microscopic level of either subthreshold or low-lying levels of inflammation within the olfactory epithelium, which can induce cellular damage as well as eventually either kill off the sustentacular cells or it can kill the olfactory neurons. We don't really have a good idea of how this process gets started and then what happens after that point that leads to the anosmia. Hypotheses related to taste are even less delineated compared to loss of smell. There are a couple of ideas out there, but it's just simply not as studied as the loss of smell. One of which has to do with hyposalivation and potential infection of the COVID-19 virus in the salivary glands, or overall a decrease in saliva content. Now, if you decrease saliva content in the mouth, that's going to decrease your ability for tastants to kind of swirl around and make contact with the taste buds. There is a hypothesis out there that similar to the anosmia in the olfactory neurons, there might be disruptions in taste bud signaling or a flat-out obstruction of the taste bud by inflammatory processes or some other obstruction that keeps the tastants away from the taste bud itself. Also similar to anosmia, there's thought that there could be infection of support cells. Just like the sustentacular support cells in the nose, there are support cells for the taste buds as well. But generally, most widely believed is we just simply don't understand why we get a loss of the sense of taste. In other viral situations with upper respiratory infections, most of the loss of taste that's associated with that is due to a loss in smell. In that same pairing of correlation of loss of smell and loss of taste, just it's not as clean in the COVID-19 infection process as it is in other viral processes where we have identified loss of taste.